Thanks. Next, we have Elisa Waxman from CHOP, or CHOP?
Yes.
Yeah, all right. UPenn, CHOP, yeah. Who's going to describe a assay to look at the different STXBP1 variants.
Great. And how do I, oops, as I drop? Okay. Green, okay, perfect. For us short people. Good morning, everyone. I'm Elisa Waxman. I'm the neuroscience leader of the Stem Cell Core in Children's Hospital of Philadelphia, and I am a member of the END team, which is a CHOP and UPenn cooperation. Just so you know, I will not be talking about stem cells today or stem cell models. Instead, I'm going to tell you about a dual-readout fluorometric assay that we developed to look at functional variants of STXBP1. Now, before I get started, I would like to thank this wonderful team of individuals who have both supported and have contributed to this work. We have both a basic science team and a clinical team who have worked together to make all of this happen. Now, there is an unmet need to identify a scalable assay to determine STXBP1 variants and determine whether or not they're pathogenic. Approximately 50% of STXBP1 variants are missense variants, many of which are de novo. Also, over the course of time, there's an increased amount of variants of uncertain significance that have been identified, increasing the need to identify pathogenicity of STXBP1 variants that could lead to STXBP1-related disorders. Now, for the creation of an assay, I took two separate premises. One, that a primary function of STXBP1 is the binding to, trafficking, and docking of syntaxin. It can be syntaxin 1A, syntaxin 1B. We've also heard about other syntaxins. For the purposes of this, I have used syntaxin 1A. Disease variants also have been known to result in protein instability and/or decreased binding to syntaxin. Taking this in mind, looking at the crystal structure, so this is a crystal structure of syntaxin with STXBP1. At the far left, we have highlighted syntaxin, and I have an arrow in, I don't know if you could see it too well, the STXBP1 that is in the background. And where I have the arrow is at a loop of STXBP1 that is in close proximity to the N-terminus of syntaxin. So I created a split neon green assay where the majority of the split neon green, so the first 10 beta sheets of split neon green, are attached to the N-terminus of syntaxin. And then in the center, here we've highlighted STXBP1, where I've attached the 11th beta sheet of syntaxin, of neon green. And so when syntaxin and STXBP1 come into interaction, it completes neon green and will fluoresce green. In addition, I've added to the C-terminus of STXBP1, M-calama-1, and that fluoresces blue. So we have a blue fluorescence, which will identify the expression of STXBP1, and we have a green fluorescence that will measure the interaction of STXBP1 with syntaxin. Concurrently, then, we can look at both the stability of STXBP1 and the binding of STXBP1 with syntaxin. Now, this is the workflow of my assay. It's pretty straightforward. I created plasmids of these two, such that we have a plasmid of the neon green with syntaxin, and then that 11th beta sheet of STXBP1 with the C-terminal tag of M-calama-1, and then I add in STXBP1 variants into the STXBP1 plasmid. Using these vectors, I transfect HEK293 cells, and a day later, they're fluorescent. We have M-calama-1 blue fluorescence that looks at STXBP1 expression, and neon green, which looks at the interaction. Then for quantitative assessment, I load them onto a flow cytometer. Now, the great part about flow cytometry is you could get a quantitative assessment of both the blue and the green fluorescence simultaneously. At the upper panel, looking at M-caloma-1, if we omit the M-caloma-1 from the C terminus of the STXBP1 plasmid, we get these gray negative cells. Using the gray negative cells, we can then gate where the positive cells would be. Similarly speaking, if we look at the neon green 2, which is the bottom portion, if we omit just that last beta sheet, the 11th beta sheet from the STXBP1 plasmid, we then get only negative cells, and these are our gray cells. Similarly, we could then gate where the positive cells would be. When we have both complete plasmids, we then get both M-caloma-1 and the neon green, and we can gate to see these positive cells. Using the assay, we looked at the M-caloma-1, once again, for expression and stability. I apologize that I keep repeating myself, but it makes it easier for understanding the assay. And then I select just the positive cells, which are our transfected cells. I do this to reduce experiment-to-experiment variability, and also I don't really want to analyze cells that were never transfected in the first place. And then we could look at the neon green and look at the mean fluorescence of both of these channels. And as you might imagine in this lower panel, as you have an increase of caloma-1 expression, you would then have an increase of neon green 2 expression. And so we assay by flow cytometry on a cell-by-cell basis and come up with a complete profile. Now, the other thing that I'm going to introduce to you is that syntaxin exists in both closed and open conformations. And these can provide functional insights into STXBP1 binding. Now, closed syntaxin is the predominant syntaxin that binds STXBP1 because it's very tightly bound. It has a high affinity for STXBP1. And in this conformation, it's soluble and it can traffic, STXBP1 traffics syntaxin to the plasma membrane to allow for synapse formation. When at the plasma membrane, the syntaxin then forms an open conformation. And this open structure occurs with binding of MUNC13. It has a much lower binding affinity for STXBP1, and so therefore it can dissociate from STXBP1 as part of neurotransmission. Now we can model the closed syntaxin with a C-terminal truncation, and we essentially remove that plasma membrane binding domain, and then the predominant syntaxin is soluble. To model the open syntaxin, we introduce 2 amino acid changes as indicated here. Now, looking at the differences of the wild type versus open and closed syntaxin within our assay, as you might imagine on the far left panel, there's no change in caloma-1, so there's no change in STXBP1 expression. However, when you look at the mean neon green channel, we have a much higher amount of fluorescence with closed syntaxin. As I indicated, it's much tighter binding with closed syntaxin versus open syntaxin, which has a reduced fluorescence and therefore reduced binding. We can actually look at these under the fluorescent microscope too. And what's really interesting about it, and I've blown this up in panels, is that with both wild type and open syntaxin, you can actually see this interaction, this neon green, at the plasma membrane surrounding these cells. Versus with the closed syntaxin, as expected, it's entirely cytoplasmic and soluble. Now, to move forward in our assay, we then took variants that are known to have certain deficiencies. So these are previously published variants. The E59K variant has decreased binding. The C180Y variant has both decreased binding and decreased stability. And then this double mutation prevents binding. Now, this double mutation, it doesn't exist in real life, but for the purposes of validating our assay, it is a really useful double mutation. Now let me orient you to this graph. This is how I'm going to be showing the data from here on out, with the STXBP1 variants at the top, the syntaxin conformational variants on the y-axis listed on the right, and then looking at mean fluorescence units as normalized to wild-type STXBP1 per syntaxin variant. So the wild type will always be about 100% because that's what we expect. The only reason why we have some dots above or below is if I put more than one wild type in an experiment, I then average them. And then looking at the different CALMA1 levels and the neon green levels, I've color-coded it so CALMA1 will always be blue and neon green will always be green. So, as we expected, the E59K variant has a decreased binding. It has very little change in CALMA1 stability. And so this is what's expected. Now, what wasn't expected is when we looked at the different syntaxin conformations, is that there was very little change in binding when looking at close syntaxin, but very dramatic loss of binding when looking at open syntaxin. This identifies for us that there are differences in specific binding conformations of STXBP1 with syntaxin for some of these variants. Looking at the C180Y, where we expect decreased stability, you can see that the level of mCALMA1 is greatly reduced with all of the syntaxin variants. Further, not only that, but we also have a further reduction with neon green. Now, I'm going to describe to you that if we have a reduction in mCALMA1, let's just say to 50% of that of wild type, and let's say the neon green was similar to that 50%, we would then attribute that reduction of neon green to that of reduced protein expression. So the fact that the neon green is even further reduced beyond that 50% indicates that it's a reduced binding and a reduced stability with this variant. Now, looking at that double mutation, there's an even further reduction of binding with the M-neon green as expected. We also identified, which previously was not reported, that this double mutation does have reduced stability. Now we wanted to look at a variety of different variants in our assay. We wanted to test benign population variants. We wanted to test pathogenic known recurrent variants, and we also wanted to test atypically mild variants. Now, these variants were provided with our clinical team with many of these variants that they actually have seen as patients in the clinic. Now, looking at the benign variants first, you can see that there's really minimal change in the CALMA1 or neon green, suggesting that there's no change in stability or binding with these variants. The only exception was the R100Q variant, which had a slight reduction in binding. However, you might imagine that there could be a slight reduction in binding, but nevertheless enough STXBP1 binding syntaxin that it would actually be still working normally. Looking at the pathogenic variants, all of them had reduced stability. So all of the mCALMA1 values are significantly reduced. However, there's varying changes in the neon green values. Most dramatic is looking at close syntaxin, where some of the neon greens equal that of the CALMA1. So as I described before, in these cases, any reduction in neon green is actually reduced stability, not reduced binding, versus, for instance, the R190W and the P480L, those have significant additional reductions in binding beyond that of reduced stability. And then for all of these recurrent variants, all of them have both reduced stability and reduced binding when looking with open syntaxin. Now, the atypical variants, those are the interesting ones. The R227S variant, this is a very mild case. In fact, this patient never had seizures. And you might imagine, looking at this profile, that it's very similar to what we just observed with the benign variants. This is different than the other atypical variant shown here. The E283K, that shows a reduction in stability, but not as much reduction in binding. In fact, with wild-type syntaxin, no reduction of binding at all. The R477P, though, that showed a reduction of stability similar to that of our recurrent pathogenic variants, however, an increased binding with syntaxin. And then the G543V variant, that one actually looks like our recurrent pathogenic variants in their profile. In case anybody wants to see them all together, this is how we can actually see the differential between the benign, pathogenic, and atypical variants that we assayed with this assay. Now we have clinical scores that were provided from our clinical team. The clinical scores are assessments of motor, communication, and vision domains. And each one of the patients seen in the clinic were scored. Just so you know, we had, for instance, multiple R551C patients, and all of them were assessed. Janine then went ahead and created a support vector machine learning model for classification of the variants. Here we plotted the neon green versus the CALMA 1. And you can see in that upper right-hand corner, all of the benign variants are clustered. And it's clustered with that one R227S variant, the one that is very mild, versus all of the other variants are on the other side of the decision boundary line. Then we were able to create a correlation between the clinical score provided, the SCSA score, and the signed distance to the decision boundary, identifying whether this patient is a benign, pathogenic, or atypical patient. So in conclusion, we've developed and validated a scalable experimental assay to assess STXBP1 stability and binding. This assay successfully identifies stability and binding deficits, and it can be used to identify binding differences even between conformational syntaxin variants. This assay may be used to assist with classification of new VUSs and provide information as to specific deficits, which actually may help with future targeted treatments. And I'd like to thank you, and once again, thank the team of members that have worked with me on this. And I'll take any questions if you have it.
Elisa, thanks so much. Great work. Really nice results. Obvious question is, did you check for functionality of these tagged proteins? So their stability and functionality in rescuing the null mutant, for instance?
So in this work, it was all done in a 293 cell assay. So it was very quick and dirty of essentially just seeing if we have binding and just seeing if we have expression. So to go further, one might imagine you would want to do it within a neuron or some sort of functional cells. So we don't have any of that concurring with this data right now.
Maybe one other question. So in your HEX cells, I noticed that in the closed syntaxin-MUNC18 interaction, you showed cytosolic expression. Could you comment on that? Did you expect that?
Actually, it is expected because closed syntaxin is predominantly cytosolic. And in addition to that, we actually truncated off the plasma domain. And so it's not going to bind to the plasma, and it is expected. I mean, these sort of variants of the closed and open syntaxin are from literature, and so they've been extensively reported on as to what's expected. So we were expecting that. What I wasn't expecting is that we would see the plasma membrane binding so nicely with the wild type and open syntaxin. That was the unexpected part.
Great, thanks so much.
Thanks.
That was great. This might just reflect my own misunderstanding, but it feels like you have this two aspects, like the stability and the binding. And I think you were saying that first, you need stability in order for you to really assess how much more extra defect you have in binding. But isn't it in fact that STXBP1's expression itself is totally dependent on STX1A binding? So it's actually almost
I think it's the reverse. That syntaxin expression is more dependent on STXBP1 binding than the other way around. Okay. Yeah. And I mean, don't forget, for the purposes of this assay, we're doing this in a 293 cell where there's robust overexpression that I'm inducing with plasmids. So we're not really looking at things quite in physiological levels here.
Okay. Thanks.
Any other questions? All right, thank you, everyone.